Characteristics of life. Before we begin, let's talk about the difference between abiotic factors and biotic factors in an environment. So, abiotic factors are non-living chemical and physical parts of the environment that affect living organisms and the functioning of an ecosystem. Examples of an abiotics are rocks or mountains, rain, wind or air, temperature, or lava. And the living things in an ecosystem are called biotic factors, such as animals, bacteria, humans, and or plants. Now that we know biotic factors are living things, how can we identify living things? They're the characteristics of life. In the characteristics of life, there are 7 factors to include. There is movement, growth and development, responsiveness, complex organized structure, metabolism, energy acquisition, and reproduction. So what does the characteristics of life actually mean? Characteristics of life are the traits or qualities scientists consider to be traits that make up a living thing. It basically means it's what makes a living thing a living thing. And the 7 factors I have stated are the 7 factors or qualities scientists consider to be the characteristics of life. Now, let's get into detail. First comes movement. Movement is when something moves on its own accord, or violation, like this man running. Keep in mind that all living things are capable of movement, like for example, plants. They sink their roots into the ground to get water and minerals and remain to live in that one area or place. Now, remember that movement can be an indicator. It is not always true. Some living things do not move. We have growth and development. Second, the most familiar kind of living thing are plants and animals. Some of us have raised our pets in our homes and watched their growth and development. But remember, some things that are able to grow larger, that is, to grow, are not alive. Like for instance, icicles. They're not alive, but it does grow. Its developments have a rather consistent form in the shape of a cone. Because ice hardens, the icicle continues to grow longer and longer, but it doesn't necessarily mean they're developing, unlike a plant where we have seedlings and sprouting into a living thing. Third is responsiveness. There are many ways in which we encounter plants and animals reacting to the world around them. If someone hits you, you might respond by hitting them back or moving back. Or a sunflower. Or a sunflower moves towards the sun as it shines. It is possible to observe non-living things in what appears to be a response, even though it is not. For example, the increase in the length of an icicle might be interpreted as a response in which growth toward the ground occurs. Next, we have complex organized structure. Most of the plants and animals we routinely encounter demonstrate this feature well, beginning with yourself. Once again, this characteristic itself is not diagnostic for a living thing. There are many things that have complex structures but are not alive. For example, mechanical cars, creation of humans, like automobiles, computers, clocks, and television are example of a non-living complex organized structure. Next, we have metabolism. Living things carry out organized groups of chemical reactions that build up their body parts, thus sustaining them and allowing them to grow, or that break down worn out or injured body parts, as well as generate energy within their own body. So if you have food, it digests and it creates energy, which is a form of metabolism. Next, we have energy acquisition. All living things require an external source of energy, like food, to maintain themselves and carry out their activities. These living things we know only use two sources of energy: radiant energy of the sun, or chemical energy like food of small molecules. They can obtain these energy from photosynthesis or energy-rich food. Reproduction. Living things reproduce their kind, and this is a process which might occasionally be seen in a field of study. Many living things whose reproduction easily observed may not reproduce frequently enough to count on seeing it during an observation period. This characteristic is often not necessary for indication of active living things. Are viruses alive? With all the 7 characteristics of life, we can identify whether or not viruses are alive. A virus is a small infectious agent that replicates only inside the living cells of organisms. We had a group discussion about this topic, and the answer is completely up to you. I made a chart whether or not viruses are alive to help you figure out whether or not viruses are alive. So this is a yes, what viruses do have, and no, what viruses do not have. So yes, viruses can reproduce. They have DNA or an RNA. They can adapt to their surroundings, and they have an organization. But they are not made up of cells or organelles. They cannot reproduce without a host. They do not have both DNA and RNA, and they do not metabolize energy. So this can help you figure out whether or not viruses are alive on your own. Next is energy. Let's start with photosynthesis. Photosynthesis are plant cells capturing energy from the sun and convert it into food, carbohydrates. Plant cells then convert the carbohydrates into energy during cell respiration. The ultimate source of energy for all living things is the sun. In chemosynthesis, organisms use sulfur or nitrogen as the main energy source. And the equation for photosynthesis is 6CO2 plus 6H2O plus energy equals C6H12O6 plus 6O2. And these are an overview of photosynthesis, which is light energy get converted into chemical energy. Chemical energy is stored in the form of glucose. Carbon dioxide, water, and sunlight are used to produce carbohydrates and oxygen, and occurs in 2 ways: light-dependent reaction and light-independent reaction. Now let's start with cell respiration. Cell respiration are food molecules that are converted to energy. These are 3 stages to cellular respiration. The first stage is called glycolysis or anaerobic, no oxygen required. The next stage are called the citric acid cycle and the electron transport chain, and are aerobic. And this is the equation. This is the equation for cellular respiration. Now, what are the differences between aerobic respiration and anaerobic respiration? So anaerobic respiration occurs in the absence of oxygen. It breaks down food substances in the absence of oxygen with the production of a small amount of energy. Produces less energy than anaerobic respiration, often called fermentation, seen as an adaptation for organisms that live in an environment that lacks oxygen. Aerobic anaerobic requires the presence of oxygen. They release energy from the breakdown of glucose or another organic compound in the presence of oxygen. Energy release is used to make ATP, which provides energy for a bodily process and takes place in almost all living things. And this is an overview of both photosynthesis and cellular respiration. We have the sun and the water going inside the light-dependent reaction, which is the first stage, where it produces ATP and NADPH. Next, when the ATP and NADPH goes through the second cycle, which is the light-independent reactions, where it goes in a circle like this, producing PGAL twice and glucose. And then so we have the light-dependent reaction, and we see the water, and the water gets evacuated to the atmosphere, producing oxygen and then carbon dioxide and oxygen out here. And then so we can see in the atmosphere, carbon dioxide actually goes inside the light-independent reactions to help with the process. So going back to the oxygen goes over to the electron transport chain. And then let's start with the cellular respiration. We see that the glucose here, and it's the 2 ATP and the 2 NAD plus gets inside the glycolysis, where it produces 4 ATPH and 2 NADPH and 2 pyranate, where the N4 pyranate gets transferred the same place as the ATP and NADPH. The ATP gets transferred to the electron to the ATP here, and the NADPH gets transferred to the NADPH and then goes transferred to the electron train. We can see that the Krebs cycle releases CO2 and then goes back inside the light-independent reaction. And we see the H2O here is released from the electron transfer chain. So it gets evacuated, and the Krebs cycle produces, I think this is ATPTH, and enters here in the electron transport chain. And the Krebs cycle produces 38 ATP, and it also gets from the glycolysis 4 ATP and ATP there. It's an overview of it. And then the ATP importance. Oh, you can't see that. But it says, one of the importance of ATP are all organisms release ATP from the breakdown of glucose during cellular respiration. ATP powers activities of the cell and allows living things to do work. 3. ATP or ADP is a cycle that releases/stores energy for a cell process. 4. ATP is used to actively transport material across the cell membrane. Next we have ecology. Okay, so we'll start with explaining what a food chain is. A food chain is a linear network of links in a food web, starting from producer organisms and ending at apex predator species or decomposers. Species. Look closely. Notice the arrows. Arrows are used to show the feeding relationship between the animals. The arrow points from the organism being eaten to the organism that eats it. If you notice that there are different parts of the food chain, there are a producer, primary consumer, secondary consumer, tertiary consumer, and the decomposer. Let's start off with producers. Producers are organisms that get their energy from the sun. Primary consumers are herbivores, feeding on plants, any animals that eat plants. And then we have secondary consumers, are animals that feed on primary consumers. Next we have tertiary consumers, they feed on secondary consumers. And I will explain in the energy pyramid. These are important parts of the food web, but the food web is not the only ecology factor out there. There are also the energy pyramid. An energy pyramid is a graphical model of energy flowing through an in a community. The different levels represent different groups of organisms that might compose a food chain. We present losses in our stimulation because we are not able to obtain all of the energy available in the food that we eat. Some or most of the energy is lost to the environment. It is lost in the form of heat energy. In general, only about 10% of energy can be transferred to the next trophic level. So you see 100, then 10%, then 1%, then 0.1% of energy. The biomass pyramid. The biomass pyramid is each level represents amount level above needs to consume. It basically means that here is how much, and this number is how much the level above it needs to consume for here. So out of this much, the herbivores need to at least consume this much, and so on and so forth. Numbers pyramid. Numbers pyramid are each level represents number of organisms consumed by level above it. Next we have cells. And now we have the cell theory. The cell theories are all cells come from pre-existing cells. Cells are the basic units of life. Organisms are created by one or more cells. All cells are made up of ribosome, cytoplasm, cell membrane, and DNA. Now, what's the difference between a prokaryotic and a eukaryotic? A prokaryotic, there are no membranes-bound organelles, no nuclei or unicellular organisms, contain ribosomes, cell membrane, cell wall, and cytoplasm. Cell walls made of peptidology can. The examples of these are bacteria. Eukaryotics are more complex organisms, typically larger than the prokaryotic. Genetic material in the membrane-bound, many membrane-bound cytoplasm, and cytoskeleton. And examples of these are animal cells. Now let me get into the different parts of the cell. First, we have the nucleolus. The nucleolus makes ribosomes inside the nucleus, eukaryote. And then this is an example of eukaryote. The nucleus contains DNA and only in eukaryotes. Ribosomes makes proteins. Oh, you can't see that. Okay. Ribosomes make proteins, sometimes attached to the ER. So these are ribosomes. We have ribosomes, we have a large unit and a subunit. All cells, and they are found in all cells, so prokaryotic and eukaryotic. The ER, or the endoplasmic reticulum, have a rough and smooth side of it. They finalize and transport synthesis of protein/lipids, and they are found in the eukaryotic. The Golgi modifies proteins and packages materials for export. They're found in eukaryotic. Lysosomes, they recycle used/damaged structures, and they break down waste. We have animal vacuoles. For vacuoles, the definition of a vacuole is stores water, minerals, food, and waste. It's in the EU and eukaryotic and very large in plants. I drew the different cells for the animal cell and the plant cell, and where each vacuole will be found. So the vacuole for an animal cell will be found here. The vacuole for a plant cell will be here, and you can see it's larger than the one right there. Next we have the mitochondria, or aka the powerhouse of the cell. It is the cell respiration that makes ATP, or energy, for the cell, and is found in a eukaryotic. Cell wall is a thick, rigid bilayer outside membrane. Structures support plants, fungi, and bacteria. Next, we have the cell membrane. They are boundaries of cell phospholipid bilayers. They are found in all cells. Next, we have the centriole. They are animal cells that are used for cell division. Cytoskeleton are fibers that provide structure for animal cells. Chloroplast photosynthesize, make sugar plants, and some protists. and are found in eukaryotic. Now, let's talk about the difference between animal cell and plant cell. These are what each of the cells have. Animal cells have centrioles, flagella or cilia, cytoskeleton, and many small vacuoles. Plant cells, they have cell walls, chloroplasts, large central vacuole, usually rectangular-ish. And they both have a nucleus, a nucleolus, a ribosome, a mitochondria, Golgi, ER, lysosome membrane, and DNA. Let's talk about genetics next. So we have the patterns of inheritance, and the patterns of inheritance is the manner in which a gene is transmitted. There are modes of inheritance, which are the simple Mendelian, where it means we have two alleles, one dominant and one recessive, and it would look like this. Incomplete dominance, where there are two alleles, they're neither dominant, so there's one large A, and one large A with a primer. And this makes a blending effect. Codominance, we have two alleles, both dominant, and they both appear, which we both have a two A's. Multiple alleles, there are more than two alleles. Sex-linked or X-linked thing is when the gene is on the X chromosome, and a polygenic is more than one gene for the same trait. Now, important definitions are, remember, a dominant has an A here. Recessive has a lowercase a. Homozygous is when there's a capitalized A, both, or a two recessive A or two little a's. Heterozygous, where we have one big A and one little a, and that usually means they are carriers of the trait because of little a if it is recessive. Genotype, we have the genetic makeup, and the phenotype is a physical character of the trait. Gene is the region of DNA codes for the trait. Let's do some examples for modes of inheritance. First, we have simple Mendelian. We are going to cross over a blue eyes versus brown eyes. We know that blue eyes are recessive, so it will be represented as little b and little b. Brown is dominant, so that will be represented as either big B and big B, or big B and little b. In this case, we're going to cross over two little b's and two big B's, as shown here. And I indicated that blue would be here, because it is two recessive, and then two recessive brown would be brown. And if we cross them over, we would have a heterozygous trait, because we have one big B and one little b. And a genotype is usually asking us for the letters. We see that there are no dominant, homozygous dominant, which means two big letters, because homozygous means the same letter, and dominant meaning either this, because this is the only two big, whereas homozygous, and then we have a dominant, which it has to be the big B's. It's asking for two homozygous dominants, and we can see here from the results that we do not have any homozygous dominants. If we see here, if we're heterozygous, because of that one big B or A and with a little a, and we can see here that we do have a heterozygous pair, because all of these have one big and one small together. They're partnered up, so we will write 4, or you can write 100%. Next is asking for a homozygous recessive, which means two little letters. And we can see here that there are no two little letters, so that will be a 0, 2. Phenotype is asking for the physical trait. Same thing, you could use percentages if you want. So it's asking for a blue. And we can see that if you have a heterozygous trait, or allele, or trait, then that means your dominant side, which in this case is brown, will always overshadow the recessive, in this case, which is blue. and we can see that there's a big B showing dominance over the little b, and we can assume that they have a brown-eyed children. So that means we have 4 brown-eyed child, but they will be carriers of that trait. Next, we will try the incomplete dominance. We will do, remember, the incomplete dominance has a blending effect, and remember, it has a red versus pink versus white. That's the way we're going to do it. And this is our little legend, basically. And we're going to see that red equals 2 homozygous dominant, pink a heterozygous trait, and white homozygous recessive. Because homozygous dominant, big letters, homozygous recessive, little letters. And then for this case, we're going to mix or blend in red and white. Red, we know it's a homozygous dominant, so up here is a homozygous dominant. White, it is a homozygous recessive, so here we would do a homozygous recessive. Now let's look. If we cross these over, we have all heterozygous children. Same thing as this one here. And then it's asking for the genotype. Which one has a red genotype, which is RR? There we see that there are none. If we are asking for the genotype for RR, one big R and little r, it's asking for pink. We have 100% of that because they are all crossing over here. Next we have white. White is the homozygous recessive, and we have 0% of that because there are no 2 homozygous recessive here. I'm going to do codominance with you. And remember, codominance is when it's both dominant. Okay, so let's go ahead and try to do a codominance with a black and the white. Remember, black has a homozygous dominant, and white also has a homozygous dominant. So that means they'll both appear. But we can see we have a third option here, which is a checkered, which we have a black and a white color together. So let's try doing a black and white. So a black has a homozygous dominant, white as well. So we will write black and white here. And we know that when it means codominance, means that both of them are gonna be dominant, and that means they're gonna have a not blending effect, but they're gonna both appear. So we would have a picture of BW if you cross these two over. These two over are also gonna have a BW. These two, and same for the last one. And we can see here that we're gonna have a checkered pattern child, chicken, or any kind of codominance traits. So genotypes asking us if we have all black, which we do not, so we're gonna write 0 there. And it's asking us here for a checkered pattern, which we do have, and we're gonna write 100%. And we're going to ask for a pure white. We do not have a pure white, so we're going to write 0. And now phenotype, it's asking for how many pure black breeds are there. We see that there are none. White, we see there are also none. And checkered, there are 4. Now we're going to do a 6-link trait. Okay? So we're going to do a crossover between a female red, which is a fruit fly, and we know that red-eyed females are more dominant than red eyes are more dominant than white eyes. Hence why we have a big R for red and little r for white. And so the way we're going to do this is the way we're going to determine females and males. We know that females both have 2 X chromosomes and males have an X and a Y chromosome, indicating that they're males. Has an X chromosome. And remember that most traits from a male comes from the mother because of the Y chromosome and then the X. X usually gets all the traits and the Y does not. And we'll see that later. when we try to fix this. So we're gonna do crossover a female heterozygous red and a white recessive white male. So we're gonna write the female here. This is XR because it's a heterozygous. So we have one capital and one little. Now we're going to do a recessive male. So it only has one trait because it's on the X only. And then traits are only on the X's. Next, we have a Y indicating it's a male. So we both have female and a male. Now in the sex-linked traits, we're going to cross over the same way as any other Punnett square. We're going to take one. So we're going to have one big R for this one. And we're going to have one little r. Same thing here. Little r, X, then little r. Now this is the part where we're going to see the Y take place. We have an XR, capital R, and then Y by itself because it doesn't have any trait with it. So Y's are the same. Now X, little r for a male, and Y because it's a male. And as you can see, as I repeatedly stated, that Y's do not have traits, and it all depends on the mother having traits for that child. So, for the genotype, we can see from our chart here that we have a one big R and one little r, which indicates we have a female red, because red is dominant eye color for fruit flies. Little r's, we have female white, because they're both recessive. I forgot to put that down. Next we have a male red right here. And then we see here that we have a male white because it's a recessive trait. For our genotypes, we're going to write out each one of these. But to save us some time, I'm just going to write down what we have. We have an XR, then XR, then XR capital, then Y, then X lowercase r, Y. And the phenotype, we're going to ask us what we have, and I'm going to abbreviate for this. For phenotype, we have one big R and one little r, which means we have a female red. Next, we have two little r's, which we can see here is a female white. Next, we have a male red because it has a dominant allele to it. Next, we have a male white because it has a recessive trait when we know that white is recessive. Now that we're finished with the Punnett square example, let's go ahead and go with the